Okay. Well, we have just a minute or two before I actually get started with the lecture proper. So I just want to do a quick recap of everything we've done so far. So the lab 5 has been graded. It's back there. Everything's in Canvas for you guys. This week we will be moving on to the integumentary system. So now we're getting more broad in how we're looking at anatomy and physiology as opposed to just cells and tissues. Now we're going to be marrying those concepts together, and we'll be talking about organs and organ systems. So we'll be doing the integumentary system. I believe that's chapter 6 in the lab manual. And the good news is it'll be a very easy lab, really easy lab. Probably the hardest lab you've had up to this point was probably last week, as far as just the amount of stuff that you had to look at and memorize. There will be histology slides on both your lab exam and your written exam and lecture. So those of you that check the videos out, I don't know if you've noticed this, I do have links in there for you. I've actually started a series of videos on histology that I'm recording. The first video is up. That covers nervous and muscular tissue. And the cool thing is it's all pictures of the slides that you guys are using in lab. So it's not like it's just some tissue that somebody uses. It's the actual tissue samples that you guys are using in lab. So that may be helpful. And then, of course, there's some other resources up there for you guys as well. Prior to your first lab exam, because there's some really tough material, this first lab exam is going to be a tough one. It's going to include all this histology stuff. It's going to include the bone and joint stuff, or articulations, and it basically what it comes down to is there's just a lot to memorize. So what I'm doing is I'm actually replacing one of your labs the week before your test, where you will not have a lab report due that week, and you can see this on your syllabus. It will be an open lab, and I'll have all the slides out, all the samples, all the models out, and it'll be an open lab for you guys to go in and study. The whole lab will just be nothing but a big open study session for you guys to kind of give you one more chance to kind of cover things that maybe you had to rush along or you didn't quite get in the lab. So that'll give you a chance to just really solidify things in lab. So that's the plan. I know it's coming. It's not quite here, but I believe about the beginning of March is when we start rolling into our exams. And of course you'll have lecture exams as well. But let's go ahead and just do a quick recap of some of the topics that we covered last lecture. And of course, there's a really big overlap of lecture and lab. So some concepts have already been covered in lab. So we finished off the chemistry, talking about the macromolecules of life. You should be familiar with carbohydrates, lipids, fatty acids, nucleic acids, proteins, protein synthesis, amino acids, etc. Okay. And add on to these high energy phosphate molecules, or adenosine triphosphate, ATP, which I'll talk about here in a minute. So that brought us into cytology last week, okay, study the cell. I just kind of did some quick recap, and I focused on some concepts that we didn't talk about in lab in great detail. Specifically what I talked about was energy production, and I talked about the concepts of osmosis, diffusion, tonicity, active transport, which is basically it's diametrically opposed to diffusion, which is a passive process, right? It doesn't need energy for that to occur. So I'll just recap those. Let's just recap energy production real quick. So there is a limited amount of ATP that's produced anaerobically, that's without oxygen, okay, right along the cell membrane as glucose enters the cell, and that cycle where that is produced is glycolysis. And a major molecule, the glucose that comes into glycolysis, is metabolized into a molecule called pyruvic acid, or pyruvate, and then that pyruvate is shipped into what organelle? The mitochondria, yeah. the pyruvate or the pyruvic acid. Okay, 8, whenever I say something is 8, that means that it did what acids do, and that's give up hydrogen ions. So when pyruvic acid gives up a hydrogen ion, we call it pyruvate. Or when lactic acid gives up a hydrogen ion, we call it lactate. So I use that terminology interchangeably. I may say lactic acid, I may say lactate. For all practical intents and purposes, we're talking about the same thing. So this pyruvate enters the mitochondria, and there are 2 major cycles that occur within the mitochondria. There is the Krebs cycle, which is the first major one. The Krebs, or the citric acid, or the tricarboxylic acid, or TCA cycle. All 3 names being the same thing. And that pyruvate, basically what we do is we rip the hydrogens off of it, okay? And then those hydrogens with their electrons are sent over. They're attached to carrier molecules, NAD and FAD, and then those carrier molecules carry them over to the electron transport. And basically, what do we have left over after we've torn a bunch of hydrogen off of carbon? Well, we have carbons and oxygens, and that's where our CO2 waste comes from, right? So carbon dioxide comes out of the Krebs cycle. Okay, and then those hydrogens are deposited into the electron transport chain, and the electrons, the hydrogens, are actually ionized, and the electrons, the energy of the electrons, are used to power several different proteins called cytochromes, and these proteins help produce ATP. And the last protein in the cycle is something called ATP synthase, and remember I said ASE, something that ends with ASE, there's a good chance it's what kind of protein? An enzyme, right? An enzyme, yeah. So ATP synthase is an enzyme, it's a protein channel, and what happens? Hydrogen ions flow through that channel, right? And it's kind of like a hydroelectric dam. Right? You have a whole bunch of hydrogen ions piling up on one side. They want to get through. As they pass through that channel, that causes the channel to spin around. That attaches a phosphate group to ADP, making ATP. Right? And then there's another protein next to it called cytochrome C oxidase, and that is where oxygen comes in. Right? And oxygen is kind of like a molecular garbage truck. Right? You have all this hydrogen. These hydrogen ions and these electrons piling up, we need to get rid of them. Oxygen comes in, right? Cytochrome C oxidase helps combine that. That creates water molecules, and when we've gotten rid of those hydrogen ions and those electrons. And that process, of course, is aerobic. And this is where the majority of the ATP for the cells produced, right, is the electron transport, okay, specifically. So that is aerobic. Most cells need to have aerobic respiration to meet their energy demands. Some cells don't. Okay? For example, red blood cells, right? Red blood cells don't have mitochondria. They don't need to make a whole lot of energy because they don't do a whole lot, right? Really, all red blood cells do is they transport oxygen. They have a protein called hemoglobin that transports oxygen. They don't even have a nucleus. And because they don't have a nucleus, they cannot make proteins, which means they can't do much, right? Which means they have a very limited shelf life. They don't live very long. They lack the machinery, the cellular machinery, all these organelles that most other cells have. So you're having to constantly produce new red blood cells as your old red blood cells die, rupture, and lyse, and et cetera, et cetera. And you'll talk about how we recycle and get rid of a lot of that stuff from red blood cell death. I don't believe I'll be talking to you guys about this, but in next semester when you go into the digestive system, there are some very important things involving the liver and bile, and one of the most important waste products from red blood cells breaking down is actually eliminated in your bile. It gives bile that yellowish color, actually. It's something called bilirubin. Okay, so there we go. Cells that this is kind of came out of your lab, but I'll just redux this. Some cells divide more rapidly than others in the human body. Some don't divide at all, right? What are some examples of cells that divide rapidly in the human body? Huh? Okay, yeah, so specifically, which cells in your skin? What type of tissue? It is epithelial. What type of epithelial? Not connective. Epithelial. There you go. The stratified squamous, right? So you have squamous cells that are, because what happens is the outermost layer of your skin is actually dead cells, right? And they're dead and they flake off. And the deeper you go, the less dead the cells are, if you will. Mucous membranes. You have a rapidly divided cells in your mucous membranes, right? In your oral cavity and along the length of your gastrointestinal tract. One of the most rapidly dividing areas in the body is in the bone marrow, where you make blood cells, right? It's one of the most rapidly dividing areas. Okay, keep that in the back of your mind, because that'll be important here in a few minutes. Let's see, you guys okay on tonicity, diffusion, osmosis? Okay, diffusion is the movement of solute, right, from high to low concentration. Osmosis is a special case of diffusion involving solvent moving from low solute concentration to high solute concentration. All right. So what goes into diffusion? And these came out of some of the experiments we did in the lab. Well, the concentration of solute, right? The more concentrated I have, the more concentrated I have my solute, okay, the more quickly there's going to be diffusion, right? If I have, okay, say 4 particles of solute here, okay, and maybe this is my container here, and I have 4 particles of solute on this side, and then I have a container here, and I have 10 particles of solute on this side. Diffusion is going to occur more rapidly in which container? The bottom one, right? The concentration is higher, okay? The molecular weight goes into it, right? Higher or lower molecular weights tend to diffuse faster. Higher? Lower, right? Lower molecular weight. Remember when we did the experiment, we would put the drop, we did the Congo red, and the one that had the lower molecular weight diffused through the agar dish faster. And that is the case with diffusion through cell membranes. Small molecules with a low molecular weight, diffuse through the cell membrane faster, right? So low molecular weight, high concentration. Are there receptors and channels, okay? Does the cell have a special channel that allows something to move? For example, water, right? There are aquaporins that allow water to move freely through. So do you have special receptors and channels that allow solute to move in and out? Polar versus non-polar. Which types of molecules tend to diffuse in and out of our cells easier? Polar molecules and non-polar molecules?
Polar.
Why polar molecules? Okay, water is an exception, right? Because there are aquaporins, because there are protein channels for water. But what makes up the cell membrane? What is the basic component?
Phospholipid binder.
Okay, so you have your phosphate heads here, all right, and then you have your lipids in the middle, okay, your 2 lipid tails. The phosphate heads, are they polar or non-polar? They're polar, right? All right, these are all partially negative. All right, all the phosphate heads. What about the lipid tails? Are they polar or non-polar? They're non-polar, right? So let me ask you again, looking at this, is a polar molecule or a non-polar molecule more likely to get through the cell membrane? What do you think? Just generally looking at this, a non-polar molecule, because the bulk of the membrane, this thick middle part here, is what?
No.
Non-polar, right? And polar molecules are going to have a harder time getting through the center of this cell membrane, right? Does that kind of make sense? Okay, so it's very important, particularly when it comes to pharmacology, particularly when it comes to drugs, right? For example, if you guys end up in a profession where you're working in the operating room or an emergency setting, and I need to make somebody unconscious very quickly because maybe I need to put an airway into them or I need to do some sort of surgical procedure on them, okay, the kinds of medications that I'm going to give them that will rapidly diffuse into the brain need to be nonpolar medications, right? Need to be very lipophilic medications so they can get into the brain quickly and they can cause my patient to become unconscious very fast. So there are very classic medications. One of them is called propofol or dipravan. You guys maybe remember this because this was the medication that ultimately led to Michael Jackson's demise. When his doctor gave him an injection of propofol, he became unconscious and he quit breathing and likely died of hypoxia or low oxygen from not breathing. Well, propofol is highly lipophilic, so it diffuses right on through those cell membranes, right into the brain very quickly and then can act. Okay, cool. Temperature, right? Higher or lower temperatures tend to favor diffusion, question mark. Higher, right? Higher. And this explains why human beings, particularly, generally is our temperature higher or lower than room temperature? Higher. Generally higher, right? That higher temperature allows chemical reactions to occur, and it also helps when it comes to diffusion of solutes. Stuff's going to diffuse much more efficiently on a warm human. Now there are other animals that have other ways of their biochemistry is a little different, and these are animals that take on the temperature of their environment, right? Some amphibians and reptiles, insects, those kinds of things. But human beings, a lot of complex stuff going on inside of us, so we need to have a kind of a warm temperature, what we call exothermic. Exothermic animals are animals that release heat. Okay, you guys okay with that? So far, so good? Okay, so then we talked about active transport. We said active transport is not passive like diffusion osmosis, right? Active transport is transporting solutes, solvents, things like that, against the concentration gradient. Okay, so it would be like a cell transporting solute back over here where it's more concentrated. That's going to take energy to push it against the gradient, okay? So active transport requires energy, and we talked about ion pumps, for example. The most important being the sodium-potassium ATPase pump, right? And it is a pump that pumps potassium into the cell and sodium out. So it creates a gradient where you have more sodium outside of your cells and more potassium inside of your cells. And that becomes important when we talk about how the heart and the nervous system works, which we'll get to a little later on. In this semester, actually, we talked about the nervous system. Okay, pinocytosis and phagocytosis are also energy. Right, pinocytosis is literally a cell drinking. Phagocytosis is cell engulfing something, kind of eating it. And then to some extent, look at that, happens again. It's every class. I don't think we've had a class yet where, yeah. All right, everybody look in shame. All right, so to some extent, exocytosis may require energy as well, right? And even moving stuff in vesicles, moving vesicles around the cell, right? How do vesicles move around the cell? Let me throw that question out at you guys. So I have little vesicles, like maybe proteins or something needs to go. So how does it get to where it needs to go within the cell as it breaks off from the Golgi apparatus? Not outside of the cell. So there's a cytoskeleton, right? You guys remember talking about the cytoskeleton, right? And there are proteins, one of which called kinesin, okay? These are mobile. These are proteins that literally move around. And kinesins, I've talked about this already, but kinesin basically has like two little feet-like extensions that attach to the cytoskeleton. and then it kind of has a head, and it can attach to the vesicles. And then what happens is ATP is used, and as ATP becomes ADP, that energy causes those little feet, okay, of that kinesin protein to move along the cytoskeleton, and it literally just walks along the cytoskeleton and drags that vesicle with it. So that would be another example of not necessarily active transport, but of a process that requires energy transporting stuff inside of the cell. Active transport is across cell membranes is really what we're talking about there. Okay, let's just quickly review the cell cycle. Okay, so we have interphase, right? An interphase is basically the cell getting ready to divide. It has 3 components. Really, I guess there are 4. I don't really consider G0 a part of interphase because a cell that enters G0 doesn't get ready to divide, right? And that might be like a neuron, a nerve cell, right? That you don't typically see them dividing. But a cell that does divide, it will enter G1. That's the growth 1 phase. And protein replication occurs there. The organelles, okay, are copied. And then we go into the S phase, the synthesis, where we copy the DNA, right? The DNA's, you make an extra copy of your chromosomes. Then you go into G2 phase. This is where you have error correction, some fine-tuning. And then once the cell's ready to go, we enter the M phase, or mitosis, okay? And you have different components. You have interphase, okay? And what happens in interphase? The nuclear envelope begins to degrade, right? Prophase, what happens there is centrioles move, one on each side, right? And then little projections come out, okay? And then start lining the chromosomes up. Metaphase, the chromosomes are lined up, a nice straight line. Okay, anaphase, right? The chromosomes are pulled apart. Half of them go to one side, half of them go to the other side, right? And then telophase is the furrow, the cleavage furrow begins to develop, the nuclear envelope on each side begins to redevelop, and then we exit mitosis and we enter cytokinesis, and the cells properly divide. Boom, ready to go. Okay, so there's a question in your lab manual talking about cancer drugs, okay? And I believe the question talked about a drug, and a lot of people didn't get this question right. I didn't deduct points, but I'm just curious as to what you were thinking when you answered this question. So the question went something like, some cancer drugs inhibit formation of the mitotic spindle. What do you think that's going to do to mitosis? And a lot of people answered, oh, it'll make mitosis occur faster. Hmm? What? What were you thinking? And I'm not asking that to any specific person, but we know that we need to have the mitotic spindle, right? Mitotic spindle forms, and that's crucial for cell division, right? And so if we inhibit the mitotic spindle, we actually do what to mitosis? We cut it off, we inhibit it. We prevent cells from dividing. And what are the major problems with cancer? When somebody has cancer, what's going on? Yeah, cells lose their special function, right? They lose their specialization, and they divide uncontrollably. So a drug that prevents cell division might be helpful for some types of cancers, right? To prevent cell division, okay? And this is just, I just threw this diagram up here to talk about another common class of cancer drugs. And you don't have to memorize this, okay? I don't want you to memorize this. This is not in your book. Okay, this is supplemental, but I think it should take a few minutes to talk about this is important because cancer is the second leading cause of death in the United States. This is one of the most significant health problems that we face today. It is right up there with antibiotic resistance, and it is right up there with cardiovascular disease and diabetes. Okay, it's a huge problem. We need breakthroughs. We need breakthroughs in antibiotics. Okay, and I believe I talked about this at the beginning of the class, that we are rapidly approaching a time Okay, we are rapidly approaching a pre-antibiotic period of time. And anybody here taking any history classes? What in general was life like before antibiotics?
Sure.
Yeah, okay, I would, people like me, okay, would be at the end of their lifespan. When we talk about average lifespan, you know, people pushing their 40s are like, and there's still countries, I actually lived in Afghanistan for a while, and it's in the 40s in places like Afghanistan, okay, your average life expectancy. Infant mortality, okay, if you get pregnant right now, okay, and probably some of you have been pregnant, some of you have children, are you particularly worried about that baby doing okay? In general, you're not. In general, you're not. Imagine, you know, a world where maybe half or more of your your babies are going to die before they're a year old. Okay, they'll die of an infection. How many of you here in this room have had infections like pneumonia? I know I had pneumonia when I was younger. I would not be here today had I not been given antibiotics that saved my life. And there are probably a lot of you. How many of you have had an appendix rupture or something like that? Had a gallbladder become necrotic? How many of you have had just a simple staph infection? And we're rapidly approaching a time where in possibly the next 10 years, we will have no way of treating. There are currently infections that we cannot treat. They're completely untreatable. People are dying. So there are lots of problems, and so I think it just bears talking a little bit about just some of the obstacles we face. So with that in mind, I want to talk about certain class of cancer drugs called alkylating agents. And alkylating agents, believe it or not, their start, how they got started, their start was terrible. They did not come from a happy place, okay? Did not come from a happy place at all. They came from a very sad place. They came from one of the worst conflicts that mankind has ever known, and that was World War I, believe it or not. Anybody taking any history classes, World War I was perhaps one of the most horrific times in human history. Imagine people fighting basically like they did back in the 1700s, where people would just line up and shoot at each other. Really old school kinds of military tactics with the technology that we have today. And this is what we had in World War I. We had machine guns, we had war technology, but we did not have the tactics, and so the casualties are just staggering. If anybody's interested, there's actually a podcast called Dan Carlin's Hardcore History. Anyone ever heard of that? Download it for free on iTunes, and he actually talks about World War I. And it is just, I just couldn't imagine, you know, hundreds of thousands of casualties, hundreds of thousands of people dying, you know, in a few days, just staggering. But anyway, what started in World War I, the Germans began with chemical weapons. So that's where this started. And there was a particular chemical weapon called mustard gas. And what mustard gas does is it's known as a blistering agent. And what happens is, after you're exposed to that mustard gas, okay, if you get some on your skin, you develop these large blisters that fill up with this yellowish fluid. That's actually where the term mustard gas comes from. Fills up with this yellowish fluid, and then they pop, and it's like you have this massive burn all over your flesh that gets exposed to this stuff. Basically falls off. It basically destroyed, and people that get mustard gas in their eyes, imagine what would happen there. Okay, imagine if you inhaled it, what it would do to the inside of your lungs, and they realized, how does this stuff work? How is this so terrible? How does it work? Well, what it does is it alkylates. So here is actually mustard gas right here. Okay, it's what we call a sulfur mustard. I have a sulfur atom, and anytime you see a little zigzag line here, every point is a carbon atom. Okay, this is a very common shorthand way of drawing molecules in organic chemistry, where instead of writing a C, every little point is a carbon. So there's a carbon atom here, carbon atom here, carbon atom here. So you have a sulfur, a carbon bridge here, and then chlorine atoms on either end of it. So it's a pretty simple looking molecule, right? So we call it sulfur mustard. What happens is these chlorines can pop off. Okay, this one might pop off, this one might pop off. And when they pop off, they take an electron with them, so they're chloride. And so they leave this side here, whatever side they pop off, positively charged, because they take an electron with them. And then that makes this highly reactive, and that makes this molecule here react with DNA. Specifically, what these molecules do is they react with guanine. You guys remember guanine was one of the nucleic acids, right? What does guanine bind to? Cytosine, right? GC, right? So what happens is these alkylating agents, okay, produce what's called an alkyl group. An alkyl group is just a chemical group that has the formula C to the N, H to the 2N plus 1. And N just means the number. So whatever your number of carbons, so if you had 2 carbons, you'll have H, okay, 2 times the number of carbons plus 1. Okay, so if you had 2 carbons, it would be, let's see, 2N, so 2 times 2 is 4, 5 hydrogens. That's all that is there. Okay, so what happens is this alkyl group actually attaches to the guanine, and that breaks the guanine-cytosine bond. Right? That breaks that bond. Okay, and if there was a gene there, okay, for example, a gene that codes for making skin or making proteins that your skin cells need to survive, you've broken that bond, that code has been disrupted, and now that cell cannot do what? It cannot make the proteins it needs for the skin. And that's how we see that damage. Now the modern agents that we use for chemotherapy, for cancer, okay, chemical therapy, that's chemo means chemical therapy, for cancer, are not sulfur mustards, but rather nitrogen mustards. It's basically the same chemical though, okay? You've got a nitrogen atom replacing the sulfur, and then you have some R group that comes off the nitrogen. It's not that important. And you have these chlorine atoms and these little carbon bridges here, and you have a pair of electrons in that nitrogen. And so what happens is this breaks down, and this is an alkyl group right here. Okay, this is actually an alkyl group. So this chlorine here popped off as a chloride. It took an electron with it, so this part is positively charged, and it is highly reactive, right? Because it really wants to get a hold of an electron now, right? Because it lost one. And this guanine has a nitrogen here in the ring, and there's a pair of electrons on top of that nitrogen, so it's kind of a partially negative. And so this alkyl group can come in and go, hey, let me bond with you. It bonds, okay, and you've got to remember that this guanine, prior to that bonding, this guanine is bound through hydrogen bonding to cytosine, right? But once that little alkyl group comes in and goes, get out of the way, it bonds, and it now breaks that bond to cytosine, right? And now that DNA, that part of the DNA, has been destroyed. And anywhere you have this cytosine guanine, okay, these alkyls can come in and disrupt that DNA and just cause massive DNA damage, and that'll lead to cell death. So what we do is we make these chemotherapy agents that target, okay, that work like this, and it's something known as selective toxicity, okay? So some cells that aren't dividing, if the cell is not dividing, okay, that means that its DNA is wrapped up around the histones and it's nice and protected, right? And so in a lot of cases, if the DNA is protected, it's not opened up and being replicated during mitosis, then that cell is going to be relatively safe from this kind of thing happening, right? It's going to be relatively protected. It's only when the cell is dividing and that DNA is opened up and it is exposed to these alkylating agents that it's going to be at risk for being destroyed. So when you give somebody a chemotherapy drug, that chemotherapy drug doesn't necessarily care, okay, not to say that anything is conscious, but that chemotherapy drug doesn't really care about cancer cells and regular cells. It goes after cells that divide rapidly, right? What's a classic feature of cancer? Rapidly dividing cells, right? So cancer cells tend to be in cell division a lot more often than not, and so their DNA is exposed, and so cancer cells tend to be more exposed to these alkylating agents. But we just got done talking about other cells in the body, right? We just got done talking about other cells that divide quickly. What are those other cells that divide quickly, that are non-cancerous potentially? Bone marrow. Digestive. Okay. And where do we see a lot of our problems in cancer patients? What are some common things that you guys have heard about people that get chemotherapy? What are some common side effects?
Lose their hair.
They lose their hair, right? Why? Because your hair follicles are growing, right? There's constant cell division there in those follicles. So, like I said, the chemotherapy doesn't care about cancer or non-cancer. It cares about cells that are rapidly dividing. You've got rapidly dividing cells here. What happens? Hair falls out. What is another common side effect of cancer drugs?
Like your skin got to protect it real good.
Possibly. But let's think real common. What are some common things that you guys may have heard?
Sick.
They get sick. They
Weak.
throw up a lot, right? Right? Nausea and vomiting, very common, right? Very common. Okay, the gastrointestinal cells are going to take a hit. And most importantly, and this is the big problem with chemotherapy drugs, one of the biggest problems, what other cells? There you go, right? Your bone marrow. And where do most of your immune cells come from? Bone marrow, the white blood cells, right? Your bone marrow, stem cells in your bone marrow divide very rapidly. And so every time you hit somebody with a chemotherapy drug, you just obliterate their immune system. Just obliterate it. And what we see, and those of you guys that go into clinical careers, you're going to see people with cancer because it's so common. What do a lot of cancer patients end up dying of? Infections. They get an infection because their immune system was obliterated. Okay, so this is the problem we face in treating cancer. How can we be more selectively toxic? Okay, how can I produce an agent that is more selective to cancer cells and less selective to immune cells and cells in your respiratory cells here, right? How can I be more selective? That's one of the big questions, one of the big problems we face. And one breakthrough is what we do is we make what are called antibodies. We make antibodies, and this will become more, you'll understand this more when you cover the immune system, I think next semester. But we can actually manufacture antibodies that are specific to cancer cells and not other cells. So the antibody will only attach to the cancer cell, and the antibody will go, okay, immune system, kill the cell, kill the cell. And there is a drug, it's called a monoclonal antibody. It's very effective. It was a big breakthrough drug in a certain type of cancer, a certain kind of lymphoma. And this drug is called Rituxan. And so you can actually, there are people that have this specific cancer where they can get this Rituxan, and some of the side effects aren't quite as bad as some of the other more conventional chemotherapy drugs. Now, radiation will also do this. Radiation is also very good. High energy light, because that's all radiation is, right? It's light. It's high energy light. Gamma rays, X-rays. Okay, high energy light. High energy photons are very good at breaking DNA up as well. And in fact, some people call these alkylating agents chemical radiation because they have very similar effects. Okay, so there we are. So that pretty much finishes up cytology. So now what I want to do, and you guys have had an introduction, which is a good thing, is you guys good with this here? Phyresis, is it going to hurt anybody's feelings? No? Okay, cool. All right. I'm actually recording this as well, and I don't know if you guys have noticed, but I'm currently up to date. So the lecture last week, I have to break them into 3 parts generally, but all 3 parts of last week's are up, and then all 3 parts of the week before that are up as well. So generally takes me about a week to get everything up. So hopefully part 1 and maybe part 2 of today's lecture will be up by the end of this week. Okay, so now what I want to do is I want to talk about histology, and we've already had a good introduction to histology in the lab. But let's just go ahead and review basic concepts. Okay, so what is histology? When I say histology, what is that?
Study of tissue.
All right, good. All right, what are tissues though?
Groups of cells.
So yeah, more than one cell doing what? Yeah, they're working together, they're working in concert to perform what?
Specific function.
Yeah, to perform specific functions or tasks, we'll say. All right. And then 2 or more tissues make up organs, and 2 or more organs make up organ systems, and 2 or more organ systems make up the organism. Okay, cool. All right, so let's start there. So there are approximately 210, I believe, about 210 in different cell types. Every 200. Okay, so 200, 210, you know, right in there. I believe it's a little more than 200, but yeah, 200's fine. Every cell has a very special function for the most part, and so it's tough, right? We can't memorize all 200. We just can't. So what we'll do is we'll talk about tissues and about cells coming together to perform functions. So let's start broad first of all, okay? Let's start broad and work our way to more specific examples, and that's of course that deductive reasoning we talked about, right? So we'll do deductive reasoning today. So broadly speaking, what are the 4 major tissue types?
Epithelial.
Okay, so epithelial.
Connective.
All right, good. Epithelial, connective. What else? Muscle
And nervous.
and nervous. Excellent. All right. These are the 4 main tissue types. So epithelial tissues. What's the major role of epithelial tissues? What do we see them? What do they do in general? Nothing specific, but in general. Covering and lining, right? Covering and lining. Okay, they line surfaces. Okay, they line the outer surface of our body, but they also line inner surfaces, right? So we see them line our gastrointestinal tract, our respiratory tract as well. Anywhere you have a surface, you're going to have epithelial tissue. You guys okay with that? Epithelial tissues also form glands, okay? They also form glands. And we'll talk about glands in a little more detail here in a bit, okay? Specifically, they form glands of the what? The endocrine, or the endocrine, and the exocrine, or the exocrine. Glands of the endocrine and exocrine systems. All right. Now, we need to know the difference between an endocrine and an exocrine gland, so let's just cover that. An exocrine gland, okay, when we talk about exocrine glands, these are glands that secrete their substances out onto a surface generally. So the classic example would be a sweat gland, right? When you sweat, right, that sweat is released out onto the surface of your skin, right? That's what an exocrine gland, or exocrine. Endocrine glands secrete, basically, they secrete into what's known as the interstitial space. And what is interstitial space? That's the space Yeah, that's the space in between cells. So let's say I have cells here, okay? And these cells are secreting substances. Generally, these are what we call hormones. And they secrete it out into the space in between them. Okay? And eventually, those substances get into the vascular space or the circulatory system, and then they are circulated throughout the body. Does that make sense? You guys okay with that? That is an endocrine gland. So it secretes its substances, hormones generally, into the interstitial space. And interstitial space is space in between the cells. Okay? And then that hormone diffuses out and eventually diffuses into the blood vessels. It gets into the blood and then it's circulated to wherever it needs to go. You guys cool with that? That is endocrine. Endocrine versus exocrine. Excellent. All right. Connective tissues. What's the deal with connective tissues? What do? Support, protect, what else? Transport. All right. They're responsible for transporting stuff around our body as well. Okay. So supporting our body, protecting our body, transporting stuff around. Okay. Huh?
It's also stores.
And storage of certain things. And storage of certain substances. Fats, fatty acids, for example, would be an important type of storage. Okay, muscle. What do muscle tissues in general do? Movement, yeah, movement, locomotion, okay? So anywhere you have movement in your body, okay, be it a movement of my appendages or be it movement of organs and other tissues that need to move, okay? Blood vessels, for example. Blood vessels can constrict and dilate, right? They can constrict and dilate. Organs, right? Your esophagus, your intestines, your stomach, okay? They move as well, right? Your pupils, right? Your pupils constrict, dilate. There's movement there as well. All right. You have your air passages, your bronchioles within your lungs can constrict and can dilate. So anywhere you have that kind of movement in your body, you're going to have muscle tissue. Cool. And then finally, nervous tissue. What's the deal with nervous tissue? Yeah, so these guys run the show, right? And the big thing with nervous tissue is they receive information, they process it, and then they send information out. Okay, so they receive, process, and send. You guys cool with that? All right, good. Now, when we talk about the general characteristics of each of these, okay, there are some key phrases that you need to associate with these. So let's talk about epithelial tissue first. Okay, so you should be thinking protection definitely. Okay, you should be thinking protection. You should be thinking about permeability, stuff getting in and out of the body. Okay, so you should be thinking permeability, which means diffusion, right? Okay, so epithelial tissues are important controllers of diffusion, stuff diffusing in and out of the body. Wherever you have diffusion occurring, you're generally going to have some type of epithelial tissue. Okay, epithelial. Why did I put this under connected? Why didn't you guys call me out on this? Right? No. Right? Permeability, diffusion. Okay, epithelial tissues. You guys got to call me out on this kind of stuff. It's okay. I won't get too angry. I'm going to go home and cry about it, but it's all right. All right? It's all right. Okay. Permeability and diffusion's a big one. Okay? With these epithelial tissues. Another thing that we need to be very concerned about with the epithelial tissues that you need to focus in on is that they are avascular. Okay, these tissues tend to be avascular, which means what? What do they lack? They lack a robust blood supply. Okay? These tissues also have what's known as a basement membrane. A basement membrane. So that means that epithelial tissues are attached, right? They're attached. Okay, they form surfaces. And so I'm just going to draw a generic surface of tissue here. Okay? And I have the actual surface here. Okay? So this is maybe outside of the body, or this is inside of the gut, or inside of the lungs, right? Okay, whatever surface here. And at the base of them, they're attached to other tissues. Okay, maybe it's muscle, maybe it's, a lot of cases it's a connective tissue that they're attached to. And the membrane that attaches them is called the basement membrane. What is the other term for basement membrane? The basilar lamina. Okay, the basal lamina. Okay, is a synonym for basement membrane. It means the same thing. And so there are two surfaces when we look at epithelial tissues. You have this base that's attached to the basement membrane. This is what we call the basal surface. And then the surface that is the actual surface that interfaces with the outer environment is known as the apical surface. So apical and basal. And just remember, basal surface is toward the basement membrane. Okay? And are these cells packed tightly together, or are they packed loosely? If you had to guess. Tightly. In general, epithelial tissues, the cells are packed tightly. Right? Because what's important? Diffusion, right? If the cells aren't packed tightly together, then if they're loosely packed, diffusion is going to be a lot easier, right? And we just said that these epithelial tissues are very good at regulating, the movement of diffusion. So they are also tightly packed. All right, tightly packed. All right, so epithelial tissues, okay, covering and lining, control diffusion, permeability. They're avascular, they're tightly packed, they have a basement membrane, and they have an apical and a basal surface associated with them. You guys cool with that? Let's move on to the general characteristics of connective tissue. Okay? Connective tissues are never exposed to the outer environment. Okay? These are never, so epithelial tissues are exposed to the outer environment, but connective tissues are never exposed. Okay, so connective tissues are always going to be deep compared to epithelial tissues. Okay, now there are some epithelial tissues that may be deep as well, but okay, connective tissues are never exposed to the outer environment. Okay, connective tissues may or may not be vascular. Some connective tissues are highly vascular. Some connective tissues are avascular. Okay, so you can't really use vascularity as a way of defining connective tissues. Okay, but most connective tissues, okay, have an extensive what's known as an extracellular matrix. Okay, which also means that do they tend to be tightly or loosely packed? Loosely packed. So here I have my epithelial tissue. This might be an example of connective tissue. I'll look at a slice of connective tissue. I have this extensive extracellular matrix, and then within little carved out lake-like areas, I'll have the individual cells, maybe 1, 2, 3 cells, okay? And what are these little carved out circular structures known as? These are known as lacuna or lacunae when we talk about more than one, right? Lacunae. The cells themselves are in the lacunae, and then they are immersed or surrounded by this extensive matrix. And this matrix tends to contain lots of fibers, okay? Some types of matrix contain more of one type of fiber and less of another, and more ground substance and less ground substance, but in general, there are 3 general fiber types. There are collagen fibers, right? These tend to be really tough, strong, okay? What other fibers are there? So you've got collagen. Reticular, those are thin filamentous-like fibers. So you have collagen. This is the real tough stuff, right? Collagen's tough. Reticular is not very tough, very thin. And what else? And elastic fibers. And these are fibers that can stretch and then go back to their original position, okay? And so you will have varying degrees of collagen, reticular, and elastic fibers embedded in this matrix. And in some cases, you will have very, very little of this. For example, blood, right? Blood is nothing but blood cells embedded in a liquid matrix known as plasma, right? So there's not going to be a whole lot of these fibers in blood plasma. There'll be other things, but not those fibers. You guys okay with that? Okay, so these are very characteristic things. You would not see a lacunae or lacuna with epithelial tissues. You guys cool with that? Okay, let's move on to muscle. What are classic defining characteristics of muscle tissue? Well, they're contractile, right? That's big. They're contractile. And going on along with the form, structure, and function, right? Structure informs us to about function. These contain specialized contractile fibers known as actin and we talked about this in lab. It's what produces striation patterns in some types of muscle tissue. Actin and myosin. Actin and myosin. Yeah, actin and myosin fiber, those are the actual contractile components of the muscle tissue. Okay? And if you had to guess, is muscle tissue going to be vascular or avascular? Vascular, right? Why do you suppose it needs to be highly vascular? Well, what do muscle cells need a lot of? They need a lot of energy. They need a lot of ATP. They need to make lots of ATP. They have lots of mitochondria. And what do you need to make lots of ATP? You need lots of oxygen and glucose, right? And where does that oxygen and glucose come from? The blood supply, right? So muscle tissue has a robust blood supply. It's very vascular. Does that make sense? Okay. So it's highly vascular. All right, cool. And then finally, nervous, and then I'll get you guys on a break. Nervous tissue, like we said, information processing, and it has these star-shaped cells, right? They kind of have a star shape to them, known as the neuron, or neurons. And these are the functional units, right? These are the units that actually receive, process, and send the information out. But those neurons can't do their job without a whole lot of support, right? And there are support cells around the neurons known as the glial cells. The glial or neuroglial cells, and those are the support cells. Okay. One more thing I'll just mention, and then we'll get you guys out of here for a little break, and that is a term. anytime you have a physical barrier in your body, okay, these physical barriers tend to involve what types of tissue, if you had to guess? A lot of cases, your physical barriers are going to be what? What kinds of tissue?
The neural,
Epithelial, right? Because when you have a barrier to the outside, you have a barrier to something, right? Yeah, epithelial tissues. So epithelial tissues and some connective tissues are involved as well. Whenever you have a physical barrier, okay, there is a special term for that barrier, and that is known as a tissue membrane. I'll write that over here. A tissue membrane. All right, a tissue membrane. These are physical barriers. Okay. We have three types of tissue membranes. Okay, we have what are known as serous membranes or serous membranes. I believe this is toward the end of your chapter. Am I right? Maybe toward the end of your chapter. That's serous membranes. That's why it's just so important for you guys to do your reading before lecture. That way you can reference this stuff. Is it the beginning or end?
131.
Is that end or beginning? I don't know.
Middle.
Well, anyway, it's in there. But serous membranes, okay? Serous membranes line internal surfaces of the ventral cavity, okay? Which is here, right? So they line the internal surfaces of your abdominal cavity, your thoracic cavities, okay? So these, when we talk about parietal and visceral layers of your abdominal membranes, right? We're talking about serous membranes here, okay? So they line the ventral cavities. They produce a lubricating fluid known as serous fluid, okay? And that helps these membranes slide around, okay? Reduces friction, okay? Also protect. And serous membranes have two components. They have a parietal component and a visceral. And the parietal component points away from the organs, and the visceral component points toward the organ, right? And we talked about this basically in the first, in your first lab, the first day of class. So those are your serous membranes. You also have mucous membranes. Okay, and mucous membranes produce mucus. How about that, huh? And mucous membranes line passages that communicate with the outside. Okay? So whenever you have communication with the external environment, serous membranes are inside, right? The ventral cavity, that's inside of your body. Okay, mucous membranes line the respiratory tract, the GI tract, okay, your mouth, the vagina as well is going to have a mucous membrane as well because it communicates with the external environment, right? All right. And then we have what are known as cutaneous. What are cutaneous membranes? Skin, yeah. This is your skin. And then finally, we have a deep membrane known as a synovial membrane. And where are they located? What do they line?
Joint.
Specifically,
Joint.
the joint.
Something that holds something too close together.
It's not the joint itself. It's not the literal joint, but it's the joint
Cavity.
capsule or cavity, yeah. So they line the capsule, they encapsulate the joint, right? They encapsulate the joint, okay? And then you've got, if you've got your bones coming in, your bone ends are covered with cartilage, right? This is the actual joint here, right? And then that joint is encapsulated, okay, in that capsule, okay, and then the different cavities within that capsule, as you said, those different cavities that segregate the different parts of the capsule are lined in this synovial membrane. Does the synovial membrane produce a fluid or no? Does. It produces a lubricating fluid known as synovial fluid. Okay. All right, get out of here. I'll see you guys in about 5 minutes, and then we will start talking about the tissue types in more detail.